Hi everyone. I'm Chen Kai Lok. You can call me Cole. I'm the application scientist from VectorBuilder. So today I'm gonna talk about accelerating AAV-based gene therapy drug development. So from the perspective of a one-stop shop experience provided by VectorBuilder. Okay, let's begin. So today, this is the overline for today's presentation. So I'll basically talking about the landscape of AAV-based therapy currently in a global perspective, and then I will talk about how vector builders provide a one-stop shop end-to-end solutions to scientists, to researchers, by offering full spectrum offerings for research and development, CRO service, including, for example, AAV capsid evolution and biodistribution analysis, and also CDMO service that is about large-scale manufacturing platforms. Okay, so first of all, about the landscape of AAV-based therapy, I would love to do a little bit of basic introduction about gene therapy and gene delivery. So what is gene delivery? So now that we have, almost a century of background of basic science research and discovery, we know that genetic materials, they are DNA and RNA, and they encode for genes, and also non-encoding sequence also help regulating our gene expression. So now that we have a more comprehensive understanding of what genetic material is, we want to apply them in different applications, different field, different areas, such as life science, research, medicine, agriculture and food, green energy, or fermentation in the industry. So how do we do that? There are different ways, different methods, or different vehicles that we can use to deliver a piece of genetic information. This includes viral vector, or non-viral vector. Recently, after COVID-19, it is very popular about mRNA with a combination with the LNP lipid nanoparticles. So delivery method can be distinguished into three categories, three big categories, including biological, chemical, and physical vehicles. So today I will be focusing a lot on adeno-associated virus, which is a trending, very popular choice among scientists who are using it to deliver genetic information as a medicine. So another basic question that a lot of people have is, why gene delivery? Why do we do gene therapy? So I would like to answer that question with a case study, a VectorBuilder case study that we want to save lives. So in last year, 2022, we partnered with Land2 Biopharma from China. We have provided a CRO together with CDMO service for about half a year's time. And together we have developed the first AAV-based gene therapy drug, LTGT06. It is to deal with a rare disease called Menkes syndrome. So this kid is about 3 years old when the time, it is really running out on this patient, on this kid. We quickly, collaborate with the hospital and Land2. We have developed this drug and be successfully applied onto this kid. So far, we are seeing a huge success on this drug, and we are really happy that we help the biopharma and also the hospital to save a life. So let's take a look an overview of all the approved AAV drugs internationally. So there are 7 AAV vector drugs that have successfully got the approval for commercialization. They are using different serotype of AAV for the delivery. Their targets are almost all about rare disease. For example, there is Zolgensma for SMA. There is also 2 AAV drugs for hemophilia A and B. Recently, there is also a drug that got approved for treating DMD. So now we have looked at the approved drug. Here is a landscape of all the publicly announced AAV-based gene therapy and also other viral vector therapies. So as I mentioned, AAV is a very popular choice right now because Right now, it is not known to be associated with any disease. So the safety profile of AAV is a lot better than other viruses that we know, and using them as a vector to deliver the gene materials. So as you can see, the green dot, the green circles here represents AAV-based therapy that is to perform an in vivo delivery directly on human. So you can see inside this landscape, AAV-based therapy is quite dominant recently. We are seeing more and more AAV-based therapy that is being developed and pushed into the clinical trials. So let's take a step back and look at the basic of AAV and understand why we are looking at serotypes, why we want to engineer an AAV, why do we use different kind of AAV serotypes. So AAV, the outer side of the AAV is formed by the capsid proteins, usually named VP123. So they are encoded by a gene called CAP. And there are 9 variable regions in the VP protein that will greatly affect the outcome and the capsid, the outer side of the AAV vectors. And this is heavily associated with tissue specificity. So 60 VPs forming an isosahedral structure. And then there are certain parts that will be having, like, for example, protrusion that will interact with the corresponding receptor. So these ultimately lead to a lot of different wild type serotypes, and they have been showing diverse tissue specificity and infectivity. They are published and studied already, I would say, very comprehensively. For example, the literature that I show here, that was back in 2012, this paper has nicely shown a biodistribution profile of different AAV serotype on mouse model. And of course, in VectorBuilder, we have also run a lot of internal biodistribution analysis on this AAV vector. So now that we know they have different specificity and efficiency, what's next? Like, people want to look at engineered serotype to increase the specific delivery, and also, you can minimize the dosage and then increase the effect. And also, there is actually another concerns about using AAV serotypes, particularly the wild type AAV serotypes. There is existing immunoresponse and also existing neutralization antibody in certain community. So seroprevalence based on regions all over the world and the serotypes, it could go up to about 50 to 70% globally. So seropositivity is actually a key factor that a drug developer should be considering in which community or like which group of patient that you will be targeting, what is the location of your clinical trial, and then what kind of serotypes that you are considering using. Do you want to develop an engineer capsid and engineer new AAV serotype so that you can bypass all this trouble? So that's why this is the trend for the in vivo vectors, the new AAV serotypes. We want to minimize immunogenicity. We want to have a much better, higher specific targeted delivery. So here is a literature that I'm citing here that analyze the usage of different AAV capsid and their frequency in global clinical trials. Of course, AAV serotypes are being used across many diverse area of disease, rare disease particularly. And we are seeing that, for example, AAV2 being one of the oldest, the early discovered species of AAV, it was used severely in early clinical trials. But of course, we are seeing a lot more new serotypes. For example, one of the famous one is 2.7MA that is being used a lot in, for example, eye disease. And we are seeing a lot more, people are doing a lot more research on developing this new capsid. So yes, new serotypes and capsid evolution, we believe that they are the keys to the future of cell and gene therapy. So now, how can VectorBuilder facilitate all this research and help researchers all over the world to accelerate a drug developing pipeline or to discover some new drug, new target, new applications? We want to provide a one-stop shop solution. So actually, our history goes back to 2014 that we first started our business. So nowadays, we have expanded our business all over the world. Specifically in Korea, we have a branch. We also have a Korean website so that we can have better communication and better service with communities, not just for Korea, but for every country all over the world. And yes, and actually last year, we have already initiated the construction of our new gene delivery research and manufacturing campus to better serve, end-to-end customer from drug discovery, research, and in development, all the way to GMP manufacturing. So this is currently our four major business segments, including research vectors and viruses, CRO services that can help optimize vectors or do screening, evolution of these vectors, CDMO services that include plasmid and viral vector or mRNA manufacturing, and of course, IP out licensing that VectorBuilder is internally working on a lot of R&D projects so that hopefully we can get more new and novel capsid or promoters that we can share and sell it to different customers. So right now, talking about our global reach, thanks to our world's first Vector Amazon online vector design platform, we have expanded our business all over the world to more than 10 branches and offices. We serve over 90 countries already, supporting more than 4,000 global institutes. We have delivered more than 1.2 million gene delivery solutions. So now let me elaborate a little bit more about our research-grade full spectrum offerings, particularly on a lot of viral vectors. So first, why did we develop our online vector design studio? So a lot of researchers should have experienced this dilemma in the lab that when they are designing a vector, it's very hard to describe. Often you have to draw something out, and when there is issues, it's hard to troubleshoot. It also takes extensive labor and time to produce this vector, no matter it's plasmid vectors or viral vectors. So our concept is that means like all of us researchers and scientists, we need a user-friendly studio that can design these vectors. We need Lego-like design and assembly so that the turnaround time of producing this vector is super fast and we are not wasting time in it, but we want to test it and apply it. We also want efficient information processing so that we can process all these components that we have in mind onto the vector and then eventually turns into a product. And that is how we have built our vector design studio online that is available on our VectorBuilder's website. And also it is linked to our scalable Industry 4.0 production model so that all this production, processing of the information that our customer designed or provided and quickly turn it into our manufacturing team. And then we can produce it and deliver in no time. So we want to make this studio like a one tool for all. We want to revolutionize the gene delivery R&D. So currently with our studio, we have already established more than 700 vector systems. So it incorporates intuitive design and also a very huge components library that currently we have built more than 400,000 components. And at the same time, we are providing a highly affordable price and also a very rapid turnaround time. So we want to minimize all the hassles that researcher has to experience when they are just simply constructing and producing these vectors. So our records is rapidly growing over the years. Based on our recent analysis, each year we design over 250,000 vectors. We have packaged 40,000 vectors a year. We have cloned 80,000 vectors a year. And also we prepare more than 60,000 plasmid DNA each year. So here are the highlights on research use AAV, since we want to know how we are going to apply this AAV in different fields, right? So VectorBuilder provide two different research grade AAV vectors. One is called research grade, one is called research plus with extra purification. For the type of AAV, we can do single strand AAV, and also there's self-complementary AAV. So self-complementary AAV, maybe some researcher is familiar with it, some maybe not so familiar. So let me explain a little bit more about self-complementary AAV. So based on the design, it comes with a virus genome that is already self-complement, so that once they are delivered into your host cell, it can take effects a lot faster than single-strand AAV, which has to wait a little bit of time before the complementary strain of DNA is synthesized. But of course, this design has limited the cargo size of the self-complementary AAV, making it the cargo size of about 2 kb. So in terms of serotypes, we can do a lot of different serotypes, including engineer capsid design that, for example, provided by customer, and all well-known serotypes, wild type, engineer serotypes. So basically, you design it, we can do it. And about packaging system, triple transfection-based transitional approach or baculovirus approach is also acceptable by us. We have experience dealing with long ORF packaging, large size, large cargo AAV vector. We have also dealt with toxic GOIs that has shown negative impact on the cells, but we already have a mature platform to deal with that. We have also successfully cloned highly repetitive sequences. And of course, we have successfully delivered this vector. In terms of libraries, some customer might want AAV library or plasmid library. We are also experienced in dealing with complex and uniform library construction. So about some research grade AAV related products, we have AAV serotype testing panel, which is very helpful when researchers having a dilemma of which kind of serotype you want to use. In your research or in your targeted disease, you have the cells and tissues, but now how do you test out which serotype has the best efficiency? So that's why we have AAV testing panel that you can customize which serotypes you want, 3 or more. It's high titer, ready to use AAV. And of course, there is also options of different promoter like CMV or CAG driven with an eGFP reporter. So we can also provide in vitro and in vivo grade based on our customers' requirement. On the other hand, we also provide AAV virus-like particle, which is basically an empty capsid of AAV. So for VLP, virus-like particles, we have ready to ship VLP that are coming from different serotypes. Also, we can also do custom services for serotypes that is not ready to ship, but we can construct quickly. And for these kind of serotypes, please feel free to contact us for these custom services. So next, let me expand a little bit into CRO, which is contracted research organization. So we provide capsid evolution, biodistribution, and as well as promoter engineering, actually. So first, let me talk about biodistribution profiling. So VectorBuilder actually has built a team that is specialized in handling with animal models and doing analysis of biodistribution. So compiling with our experience on plasmid and viral vector, we have been providing CRO service for many years already. And here is an example of like how what we can provide to the customer. Different kind of injection, local injection, systemic injection of the AAV drug, and then doing the profiling in different organs on mouse or monkeys. So now that we can do the analysis, we also provide capsid evolution. So here are some strategies that we can provide to our customers. So when we want to engineer the AAV serotype, there are different ways to do it. For example, doing a random peptide display, meaning that we are adding some random peptide on existing serotype on normally, for example, one of the hotspot for peptide display is the number 8 the variable regions VL8. So this is one of the popular choice for doing peptide display. Some others being error-prone PCR, serotype family shuffling, some insertional mutagenesis, and also ancestral sequence reconstruction. And because that VectorBuilder has been dealing with a lot of different serotypes, we can do this library construction on existing serotypes like AAV 1, 2, 8, and 9 are also some very popular choice. So after you prepare plasmid library and package the virus library, you have multiple selections. For example, you can do in vitro screening on cells, and then also, you can also do in vivo screening on mice model or monkey. And based on your application, we can screen the AAV library based on, for example, RNA transcription level, DNA, the concatemer level, or even protein expression level. So that will be very customizable by our customer. So let me give a little bit example of how and how good we can provide the CRO service. So at the very beginning, it's very critical that when you prepare a plasmid library, your theoretical composition of the codon should be your actual library should have the codons composition that is very close to your theoretical results. So in different projects, we have been providing, for example, a 7-mer peptide display library in the VR8 of one of the AAV serotype. So our actual sequencing, NGS sequencing result shows that the library is having a very close to theory composition. So once we package the virus, we can see the graph below that. We can analyze the bias during the viral packaging, for example, in a lot of different positions from the 1st to the 7th peptide. It is not in favor for some of the amino acid. However, in some position, they show different biases towards the different amino acids. So this is something that we can do to make a high quality uniform library. Then the next thing to ensure the quality of our virus library is to incorporate a certain amount of stop codon in the plasmid library so that it is to prevent cross packaging. So once that we have packaged the virus, we do the NGS again and realize that there is a depletion of stop codon, meaning that the capsid that it expressed that is containing your capsid gene is actually corresponding. so that, in the later stage of this project, you should not be seeing the press, like you should not see much of the presence of this stop codon. That is something VectorBuilder can do. So here is some case studies, some actual surfaces that we have provided, but of course we have hide some confidential information. For example, in the first case, we are helping our customer to improve a retinal discovery with peptide display. And this one is screened based on the RNA transcription level. We have successfully screened out several capsids. As you can see here, there is a dot here that has the best performance, and our customer is very happy about it. And in the second scenario, we are trying to help our customer to make a new capsid that is for liver-specific delivery. It is based on the DNA concatemer level, and it's actually very successful that we got a lot of good candidates for the customer to select from. And of course, there is more extensive proof that our capsid evolution is successful. In one scenarios, our screened AAV top candidate compared to the wild type shows a much better protein expression, which is shown by the EGFP here. So capsid is not the only thing that you can do to help with the specific delivery. You can also do, for example, engineer promoter that is only expressing or driving the gene transcription and then expression in your targeted tissue. So in one of the case that VectorBuilder has worked with, we are trying to help this customer to generate a clone-specific small-site promoter, because this customer will also want to avoid some patented promoters. So what we do is use some known clone cell specific promoters and shuffle the sequences and build a library. Here is the initial data that is comparing one of the most specific promoter ProA1 to drive the expression of the red fluorescence TD tomato. We also construct the library, a shuffle library of this promoter sequence to drive the expression of EGFP. As you can see here at the beginning, the specificity is actually not good, that there is a lot of expression of these AAV delivered genes in different parts of the retina. However, after the collection of the clone nucleus, we can do multiple rounds of screening. That is a circle of promoter library construction, AAV library preparation, and then it goes into the mice in vivo screening and ends with NGS sequencing. So depending on the result, we can proceed further, to use the enriched library to prepare another library of promoters and package the AAV again and go through the whole thing again and again, so that you can refine your results. Or if you are satisfied with your results, you can get some selected candidates. So in this case, we have successfully generated some candidates that is with a very miniaturized size, a very good and high specificity, and also it's a new IP for the customer, which is actually owned by the customer, but not by VectorBuilder, and which is very different from all the patented promoters. So this is the result, that the new promoter that we generate compared to ProA1, which is actually a 2 kilo base promoter. This new promoter is actually with a few hundred base that is smaller size compared to a lot of published clone-specific engineered promoters. And the result and the effect for the delivery is actually comparable to ProA1. And our customer was very satisfied with this new IP for the promoter. So the next thing that I want to introduce is our CDMO platform for our manufacturing, particularly for AAV. So overall, for all our CDMO manufacturing service, our capabilities lies in multiple GMP production suites. Right now, we have more than 10 of them, and the total area for our GMP facility is about 100,000 square feet. We are capable of phase 1 to phase 3 clinical trials manufacturing. We also have independent process development and pilot runs lab that is about like 8,000 square feet, and independent QC laboratories and QC team that occupies about 9,500 square feet. So thanks to our experience about viral vectors and different kind of methods for their production, we now have the technical know-how about the vector design, how we package them, how do we scale it up into a large scale bioreactor production, and also doing like mass production of some, for example, packaging plasmid. We also build an integrated platform that we have a mature processes that is for different viral vectors and plasmid. We also build in-house QC that we are going to provide validated in-house QC panels based on the projects request by customer. We also have professional teams that is doing a lot of development of the process and also researching on advanced technology. So ultimately, we want to provide a best price and also the fastest turnaround time for our customer in the industry. So this is some details about our CDMO technology platform. For plasmid, we have already revolutionized the design studio, but that is not enough. We also do a lot of development on our platform. We have a lot of way that we can optimize the fermentation to increase and boost the plasmid DNA yield. And right now, our CDMO departments have already delivered more than 150 batches of the plasmid DNA. On the other hand, our main character here today about AAV, our CDMO department has developed the process for all the well-known serotypes, and they have provided 23 batches of AAV vectors that are being used in preclinical study and human trials. So our CDMO team has already did extensive optimization of the upstream process in the suspension and also in adherent culture method. They also develop an anion exchange column strategy that can be scale up to the rapid processing of huge volume of virus harvest and separate the full capsid from the empty capsid. But of course, I'm gonna say this is not just it, because we have extensive experience dealing with AAV vector from research grade to GMP grade. We have, we never stop optimizing our AAV packaging system. We have actually engineered a lot different ways of our AAV packaging vectors. We have successfully boosted the virus yield that is on a lot of the low yield vectors such as 2.7M8, AAV 6.2. This can actually meet a requirement so that the process to scale up and everything will be a lot easier. With this platform for our manufacturing process and then engineer packaging proprietary packaging system, we already greatly reduce our cost and time that we spend on the manufacturing. And ultimately, the proposal and project that we can provide to our customer, it comes with a better price and turnaround time. So about CDMO, another thing that is very critical is the quality of the drug itself. So for AAV vectors, a lot of people are very concerned about full capsid and empty capsid and what's the ratio between them. We have developed different ways to measure the full and empty capsids. For example, analytical ultracentrifugation. We are very familiar with the analysis of the empty peak and the full peak for the capsid. We can also do TEM, the electron microscopy. We can also do the charge detection mass spectroscopy. So we are actually spending a lot of time developing these methods and validating them. And of course, we are also trying some very new technology, also called mass photometer, to do a real-time monitoring of the capsid contents. This is to monitor our effectiveness of our process. For example, the graph shown here is actually samples that is taken between each step of our downstream purification of our GMP AAV vector. So as you can see, after the pink color affinity column, we can obviously see that all the impurities have been significantly reduced already. And then after anion exchange, we have successfully separated a portion of AAV out that have a much higher full capsid ratio than the empty capsids. But of course, aside from GMP manufacturing of AAV vector itself, we understand that there are customers that want to do GMP manufacturing of the virus themselves. However, they could be in trouble sourcing a GMP grade plasmid vector for their virus packaging. So that's why we also push out this new surface. This is actually launched this year, that we provide GMP-like and GMP-grade AAV packaging plasmid along with our lentivirus packaging plasmid, so that they are available immediately. They are off the shelf. They are kanamycin resistant, so no worry about some regulatory concern of ampicillin. They are also royalty-free, and we are also preparing DMF that will ultimately simplify, for example, the FDA IND application. So last but not least, this is our vision for the cell and gene therapy. We want to provide a one-stop shop experience, starting from a very beginning, vector design and cloning, also, helping our customer to do a capsid evolution to make a better vector, and then proceed to the next step, to the process development into a large-scale GMP grade manufacturing. And for GMP manufacturing, we can do the whole set of, for example, process development, analytical development, manufacturing of the plasmid and virus in different systems, aseptic fill finish, and also the regulatory support, for example, IND enabling services, and also the documents drafting, and also providing the final CTD documents for the application for the IND. So that will be all for my introduction today. If you have any questions, please feel free to contact our office. There is an email address that is specifically for communication in Korean. You can contact our Korean office at any time. There is also our English email address that if some of you are from the English country, you can also contact this email. If you are curious about our CRO or CDMO service, you can also contact the corresponding email address for more information. Thank you very much.